going to be talking about Torreya taxifolia and the Tradekeepers project, which is an FNPS project. I work for the Florida Native Plant Society. I'm the director of North Florida programs and the Tradekeepers project manager. And this is a collaborative effort. We work really closely with multiple institutions, but especially with the Atlanta Botanical Garden. And I'm very lucky. There's the picture at the top here is my friend Ashlyn, who I get to work with. She's wonderful. She is the Gulf Coast coordinator for the Atlanta Botanical Garden, and she joins me on a lot of surveys. So to begin, I just wanted to mention that the natural range of Torreya taxifolia is found on ancestral and unceded lands of the Muskogee Creek and Apalachicola people. They, along with their ancestors, have been stewards of this unique region for many generations. And I also wanted to acknowledge the organizational and individual contributions to the conservation of Torreya taxifolia. There have been so many organizations and individuals that have contributed so much for decades to the conservation of this species, and there are just too many for me to adequately acknowledge. So this presentation represents the current work of the Tradekeepers project, which is built upon and in collaboration with the contributions of many other organizations and individuals. And so I just wanted to give credit and thanks to everyone who has contributed to the accumulated knowledge and protection of this species. Okay, so a little bit about our project. The Tradekeepers project was originally started by members of the FNPS Magnolia chapter, which is my chapter. It was spearheaded by Lee Brooks, Billy Booth, and Helen Roth, who are all local landowners in the native range of Torreya, and they're native plant enthusiasts and FNPS members. And they began surveying privately owned lands in the area for undocumented Torreya trees, and they collaborated with the Atlanta Botanical Garden, University of Florida, Torreya State Park, and U.S. Fish and Wildlife. And the project was adopted by the state organization of the Florida Native Plant Society in 2019, and I joined the project then and started working for FNPS. So currently we work closely with the Atlanta Botanical Garden. Like I mentioned, Ashlyn Smith is the Gulf Coast coordinator for them. And we are continuing to survey private and public lands, mostly private, for remaining trees. And so we collect data on these trees that we find. We also collect tissue samples for genetic testing, which the Atlanta Botanical Garden will be doing. And we collect cuttings as well for ex situ conservation to, conserve the genetic diversity of this species. And I also collect canker samples for the University of Florida for their continuing research on the Fusarium that is infecting them. And I'll go into more detail on that shortly. But first, the star of the hour, the Florida Torreya taxifolia. This individual picture here, that's me standing there with my friend's little dog. It is probably, I would say, the healthiest and most mature representative of Torreya taxifolia that's found within its range. This, although it's probably so healthy because it's a planted specimen at a state park. And first, I'm going to go over a few basic facts about Torreya, and then we'll go into what has happened to it. So, Torreya is a genus of conifers made up of 6 or 7 species within the family Taxaceae, and that's commonly known as the yew family. And 4 species of Torreya are native to Asia, and the other 2 are native to North America. We have Torreya californica on the West Coast, and then Torreya taxifolia here in the Southeast. And the genus was named after the American botanist John Torrey. He was actually living in New York, though, And he identified it because he was sent specimens from Hardy Kroom, who lived in the area, who was a kind of amateur botanist. And they are medium to small evergreen trees. They grow to 30 to 60 feet tall at maturity. And I wanted to point out the flat arrangement of the needles on these branches. They're linear kind of and flat. They're very stiff in texture, and they have a little sharp spine at the tip, which will definitely pierce you and be painful. And I get lots of kisses from Torreya. So the needle-like leaves are spirally arranged on the shoots, as you can see here, but lie flat on the branches. And again, they're hard in texture, they're stiff and sharp. So Torreya can be either monoecious or dioecious. And Torreya taxifolia is dioecious, which means that both the male and female reproductive organs are on separate individual trees. So there's either a male tree or a female tree. The male pollen producing cones are grouped in lines along the undersides of the branch. You can see a little illustration here. And the female seed producing cones are single. They are grouped in or grouped in 2 to 8 on little nuts on a short stem. And they begin small. It takes them a very long time to mature, about 18 months. And they hang from the undersides of the branches, and they produce a single large, pretty big nut-like seed covered with a fleshy outer coating. So again, notice the flat leaf arrangement. You can see the male and female. There's a female cone right there in the lower left-hand side, and the male pollen producing cone up on the upper left-hand side. Okay, so there are a couple of tree species that are commonly misidentified as Torreya, and one of them is another rare endemic and endangered conifer that occurs in the same steephead ravines as Torreya taxifolia, and that is the Florida yew, Taxus floridana. This species is possibly even more rare as far as number of individuals compared to Torreya taxifolia, but since it has always been pretty rare and it hasn't had this catastrophic decline which Torreya has experienced, it hasn't garnered as much attention as Torreya has. And from a distance or in photos, it's very easy to confuse Taxus floridana for Torreya taxifolia. But notice here is the Florida yew. Notice that the Taxus needles are shorter. They have not quite as sharp of a tip. The new growth is glaucous, as you can see, that kind of matte, waxy coating on it. And if you're able to touch the plant, then you will know right away whether it's Taxus or Torreya, because the Taxus has soft, pliable needles as opposed to the sharp, stiff needles of Torreya taxifolia. And so similar to Torreya, Taxus floridana is dioecious with male and female cones on separate plants. Here's a little comparison here between the Torreya and the Taxus. Again, the Florida yew has a soft, flexible needles. They're also evergreen, they're also dioecious. And the bark is also a good way to differentiate them as well. For photos at least. If you're in person with a Torreya or a Taxus, you can tell the difference for sure. But the Taxus bark is on the left, on the lower right-hand side here. It has kind of a dark purplish almost hue, and it's flaky, whereas the Torreya bark is not flaky and has these kind of, linear fissures in it. Okay, So here's a slide comparing the reproductive structures of Torreya and Taxus. And again, as I mentioned, Taxus is also dioecious with separate individual males and a separate individual female. The female seed cone is berry-like with a single scale developing into a soft, juicy, bright red aril that contains a single dark brown seed. And the fruit also occur singly or a few of them underneath the branches, on the underside of the branches. The pollen cones are very similar in appearance to the Torreya pollen cones. They're rounded and they are also produced on the undersides of the branches, just like Torreya. And then I often get emails or phone calls from people who say, I have found a Torreya in my yard, or my neighbor has a Torreya. And invariably, it's always this species, which is commonly confused with Torreya. And this is the Chinese fir, Cunninghamia lanceolata. This species is not in the Taxaceae family, it's in the Cyperaceae family. And it's similar, very similar in appearance. I could definitely see why people confuse it for Torreya, but there are some differences. This species is native to South Central and Southeast Asia. It's from China, obviously. They're monoecious, so they produce both male and female reproductive cones on the same plant. And they also produce these cones that look very similar to like a pine even. And its needles are much more similar to Torreya needles than the Taxus. They're also stiff and sharp. They're not quite as sharp as Torreya. They don't bite quite as hard. But the leaves are also spirally arranged on the stem of this or trunk of this species, but they're also spirally arranged along the branch. So that's one difference between Torreya and Cunninghamia, is that the spiral arrangement of the needles on Cunninghamia branches compared to the flat linear needle leaf arrangement on the Torreya. So here's a comparison of the three species, so you can kind of take a gander at them and differentiate them. You can see the Cunninghamia, the Chinese fir, has a very fluffy appearance to its branches because the needles are not arranged flat. So that's one way from a distance to be able to differentiate it, but also seeing a 30-foot tall Torreya is not likely. So it's more likely to be a Chinese fir. All right, so back to Torreya. So Torreya is endemic to the unique steephead ravines along the Apalachicola River in the Florida Panhandle, with a few populations occurring just north of the state line in Southwest Georgia. And I get to actually visit those populations this week, so I'm very excited to see those because I haven't seen them yet. Okay, so this range, their range has always been narrow. There's no published evidence that they ever had a larger range, but it was once pretty common within its range. It was a regular component seen in these steephead ravines. And there were plenty of tall, 30 to 60 foot tall mature trees that were often actually used for a variety of uses, like fence posts. People use them as Christmas trees even, for its lumber. They have very upright and very erect, straight trunks. So they were, definitely had a lot of uses. Okay, so the steephead ravines that they occur in are a really unique geological feature. And they're a large part of why Torreya is here. They are ancient barrier that Florida, I guess most of you guys probably know, the Panhandle is bisected by the prehistoric coastline. And there were a lot of these areas were ancient barrier islands or dunes. And because of that, there's a lot of sand, about 20 to 30 feet of sand, typically above the more, hard packed clay and kind of limestone soil underneath the sand. And because of this, deep sand at the tops of these bluffs, the water percolates through the sand and then feeds down and erodes at kind of midway through these slopes. So rather than a typical ravine slope or a typical ravine, you can see this aerial photo on the left. The typical ravines would end in kind of a V shape because they're eroding from the top down and carrying, it's whatever from the top down. They're eroding the soil. Whereas in steep head ravines, because of this deep layer of sand, the water percolates through and then hits kind of that hard pan, and then it kind of seeps out midway through these ravine slopes. So you'll see as you're hiking through these ravines, kind of wet areas that are seeping out water about midway through the slope. And that creates a erosion. It creates erosion that has a different look to it. So it creates this big bowl rounded shape at the end of the ravine rather than like a steep or sharp V. And it also creates incredibly steep ravines because it's kind of almost eroding from beneath and the top is slowly falling down. And so these slopes are very steep, often 45 degrees, and then they have this large bowl at the end of them, and they're continuing to move and grow. So they create their own microclimates because they're consistently much cooler, and that water is consistently running. It doesn't stop running seasonally or anything. So, and it's cool water. So it creates this nice, cool little microclimate, in the heat of Florida. And it's also consistently shadier because there are so many, trees. Well, there were before Michael. So I just wanted to add a few photos that give you kind of a feel of what the ravines look like. You can see on the left here, you can see how steep that slope is. Some of the slopes are literally like 90 degrees or even less because they're eroding in midway through the slope and there's a little ledge of soil above. But they're very steep. And then down at the bottom of these ravines are these little creeks that are created by that water that is continually flowing. And they're really lovely. Here's one. They usually have sandy bottoms because the soil from the sand that's up top on the bluffs is carried down into these little streams, and they create these beautiful sandy bottom streams. So the Apalachicola River is around 30 million years old. And some of the plant species found there are even older, including Torreya, because they were already established before, half of Florida emerged from the sea. So again, as I mentioned before, the prehistoric coastline bisects the Florida panhandle. So, north of the coast, prehistoric coastline, that feature is called the Cody Scarp. And north of it, we have like much more clayey rolling hills, and south of it, it's sandier with limestone outcropping and typically flatter. And interestingly, that Cody Scarp, that prehistoric coastline, bisects the range of Torreya taxifolia. So these species established themselves in the Apalachicola River region, and as the climate warmed over time, they persisted in these cool microclimates. And so there are a lot of species that are especially abundant in these ravines. that are common in the Appalachian Mountains. And that's because the Apalachicola River is the only river in Florida that originates in the Blue Ridge Mountains of northern Georgia. So all other rivers in Florida originate somewhere along on the coastal plain. So many of the species that occur there move down here during glaciation and have persisted in these cool ravines. So as I mentioned before, the historic range of Torreya taxifolia has always been narrow, but it was once a common component of the wooded steephead ravines found in this range. And there were plentiful 30 to 60 foot tall specimens that were frequently harvested for their durable wood by locals. However, by the 1950s, this species began a catastrophic decline. So these trees that once grew to 30 to 60 feet, it's now very uncommon, or it's rare to find one 10 feet or more in the wild. There's a photo of me on the right here standing with next to one that's probably about 12 feet or so, maybe a little bit less, maybe, no, that was a tall one. That was probably like 15 feet. So because so few trees actually reach maturity in the wild, there's virtually no reproduction. And it is considered functionally extinct because of that. And over the years, the reasons for this very rapid decline have been hotly debated, and many theories have been surmised, although habitat degradation due to development, as well as silviculture, which is the tree farming essentially, which is very big in this region, and climate changes and other human causes have all contributed to its decline. They're also susceptible to deer rubbing. Deers love to rub and rut against them, possibly because it has such a specific scent, Torreya. Oh, I didn't mention that. When you break the needles or, cut into the bark of Torreya, it has a very distinct and strong smell. And people describe it different ways, but for me, it smells like the way a tomato plant smells. If you've ever smelled the leaves or the plant of a tomato, that's what Torreya smells like to me, but like concentrated by like times 10. So they're very strong smelling, and that's possibly why deers like to rub on them. But that causes some damage, mechanical and otherwise, to the trees. Unfortunately, the primary culprit of the death and decline of the species is due to another reason, Fusarium torreyae. So this fungal pathogen, Fusarium, was identified and described by Dr. Jason Smith at the University of Florida, and his lab is continuing to research this newly described species. This species was previously unknown to science. It was never described before. So Jason suspects that this fungus evolved in Asia along with relatives of Torreya taxifolia, native to that region, and was likely introduced through the import of non-native species for horticultural uses, although, it's very difficult to know for sure. But there are many, many, many species of Fusarium, apparently. I'm learning a lot about Fusarium now. And they're well studied because they are an issue with agriculture and food crops. And this species is part of kind of a basal clade of Fusarium that has only two sister species that occur in that clade. And both of the two other sister species that are the most closely related to Fusarium torreyae occur only in Asia. They're endemic to China, actually. So that's one of the reasons that Jason suspects that this Fusarium likely evolved in China and was introduced to the US. The other reason why it's likely that it was introduced is because of the steep and fast decline of how quickly this species is going extinct. That would suggest a lack of resistance to it because they did not evolve with it. So the trees can look healthy, and especially planted specimens tend to look and grow very healthy until for some reason they're stressed. Of course, planted specimens are much easier to maintain their needs, with consistent water and fertilizer, etc. But trees in the wild tend to be exposed to more stressors. And when they are stressed, the fungus causes a kind of leaf spot, a leaf blight, and stem cankers. And it ultimately can kill the tree back down to its root system or kill the plant entirely. And the tree might resprout because the roots are still living, but it remains infected with the Fusarium. And so pretty much all the trees that are existing in the wild still are all resprouts. There aren't any that are still the same plants, the same trees that were there in 1950. It's genetically the same individual, but they're not going to be tall and mature like they once were. So, and then again, I mentioned the trees can have no symptoms of the Fusarium, but still be infected. And so all Torreya taxifolia that have been tested for this fungus have been positive for it. And it's suspected right now, it's considered that it's most likely all known material of this species is infected with this Fusarium. So that's not good. And most worryingly, the Fusarium, it's spread by airborne spores. It has been shown to infect unrelated hardwood species, as well as other rare conifers, such as Carolina hemlocks, eastern hemlocks, and other pinus species. So for this reason, it is not recommended to plant Torreya taxifolia outside of its near native range to avoid unforeseen consequences of spreading this fungal pathogen. The hemlocks are already suffering from the woolly adelgid, and adding to that, this Fusarium could be really the death knell for our native hemlocks. So again, this is the most important thing that you can get from this talk, is not to plant Torreya outside of its native range. So Dr. Jason Smith at the University of Florida has been doing extensive trials on this Fusarium. And he has done many experiments inoculating other genera with this Fusarium, and it definitely infects and kills other species. He's done, let's see, I talked with Jason just earlier this afternoon, and he was telling me about his research. So he has done 3 separate trials, and they also do like mock inoculated plants that don't have the Fusarium as like a control. And the most susceptible to the Fusarium was actually Pinus pungens, which is the photos that you can see in the lower right-hand side. In just 4 weeks post-inoculation with the fungus, it killed the tree. So that is really quick. And it infected and negatively, affected the health of pretty much all the species that he tested it on, with the exception of one of the Torreya that occurs in China. It's native to China. He tested it on two Asian species of the Torreya, and one of them had a little bit of infection, some signs of infection, and the other one had almost no signs and remained healthy. So that's another reason that Jason thinks that it's most likely that this Fusarium evolved in Asia. So oops. Unfortunately, there are a lot of people, when they hear about a rare or endangered plant, their response is to plant. They want to plant it in their yard because they want to help the plant. And, often that is useful. And unfortunately, that's not the case with Torreya. And because there have been so many different theories that have been spread about why this species is declining. It was only in 2011, remember, that they actually identified this Fusarium that's actually killing the plant. There's a lot of outdated information out there, and a lot of people are still planting Torreya outside of its native range, hoping to save the species. And there's a group called the Torreya Guardians who are attending this talk this evening as well, who are working to plant the tree further north in order to save it. Unfortunately, that's not useful. And we are concerned about spreading this Fusarium. This tree, Torreya, it's not going to go extinct because it is the most well-represented species in ex situ conservation. The Atlanta Botanical Garden has over 700 genetically separate individuals of Torreya represented and growing, in their collection. And so it's not like the tree is going to go extinct. We still have a great handle on this species. So outplanting it is actually such a great risk, and it's not necessarily protecting the species in the long run. And it's threatening our eastern forest, our eastern hemlocks and our pines, and it could be a real disaster. So we recommend don't plant Torreya. And if anybody has any questions about the Fusarium, Jason said he would love to talk to anybody, and he'd love to share his research and share his trials, his paper about all of the different species that he inoculated and did these trials on will be published soon. I think he said next month in the Journal of Plant Disease. So you can look out for that information coming out soon. So unfortunately, another thing to add on to the troubles with Torreya, we had Hurricane Michael, which I'm sure all you Floridians are very familiar with, in 2018, and it drastically changed these slope habitats that Torreya exists in. And it has also made surveying for Torreya much more difficult and dangerous, as a combination of these very steep slopes, which are often wet because they're seeping, like I mentioned, and then you have felled, dead trees all over these slopes. And then to add to that, jungle gym, you have all of the vines and secondary growth that is taking advantage of the open canopy and exposed soils. So the grapevines and the blackberries are just going wild. And the grapevines are actually the most dangerous plant in these ravines because they just, tangle you up and trip you. So surveying has not been easy. Oh, and also I should mention that about 20% mortality in Torreya has been recorded due to Hurricane Michael. So that's with a tree that's already in such few numbers and so precarious, that was a huge blow. So here's a timeline of the decline of Torreya. 1938 was when they first observed leaf loss. And then by 58, there were dead trees noticed up on the high slopes. And so that's another contributing factor to the idea that this was an introduced fungus, because it likely spread from yards or gardens down into the slope. And then 1962, all canopy-sized trees were dead. That is an incredibly fast, decline of a species to go that quickly from 1938, the first signs, to 1962, all canopy-sized trees dead. That is very fast. And again, that speaks to the likelihood of this being an introduced Fusarium that these trees had no, they had no built up immunity to or resistance to. And then in 1984, because the trees were, very quickly declining and they were potentially going extinct, they were listed as endangered under the Endangered Species Act. And then in 1986, the U.S. Fish and Wildlife Recovery Plan was written for Torreya. And in 1989, the Atlanta Botanical Garden began their conservation efforts with this species. So the Atlanta Botanical Garden has been working with the species for a very long time. And in March of 2018, the Tradekeepers was formed as an outgrowth of the Florida Torreya Tree of Life Conference. And in October of 2018, the same year Tradekeepers was formed, we had Hurricane Michael hit, which, as I mentioned, caused about 20% mortality of the species. And as of 2019, there are around 700 to possibly 1,000, and we're adding to those numbers as we continue to survey. There are about that many remaining, and the Atlanta Botanical Garden and FNPS we're continuing to survey. So it's currently considered functionally extinct because there's not any sustained reproduction in the wild. We have come across reproductive females in the wild, but because there's not a sustained reproduction, it's considered functionally extinct. Okay, so our current conservation actions, what are we doing? There are a large number of actions that multiple organizations are taking. And I can't go into all of the different efforts in detail, but I'm gonna briefly just go over all of them very quickly, the ones that we are involved with. And each of them are very important to the continual survival of the species in the wild, and provide information to help us better understand how best to protect the species and how best to treat this Fusarium. So we have fungicide treatments are underway. So Jason Smith and colleagues, they isolated Fusarium Torreya directly from cankers collected from the trees. And they found 5-year-old seedlings died within 6 to 8 weeks of inoculation. So when they inoculate Torreya even, they die very quickly. And as I mentioned, there are many, many species of Fusarium out there, and they all have different mechanisms of functioning. And so learning how this species and this individual new to science species functions will be very critical in knowing how to effectively treat it. And so I collect canker samples from Torreya when I'm surveying, and I send them to Jason and his colleagues, and they are continuing to study the Fusarium. And so again, for all questions about the Fusarium, please contact Jason Smith. He's happy to talk to anybody about his work. And his email address is below here. So fungicide treatments. Now that we know it's a fungus and it's been identified, and successfully isolated. We still don't know exactly how it functions, so we don't know the best treatment for it, but ABG went ahead and started fungicide trials to see if any of the typical fungicides that are used in agriculture to treat Fusarium might work on this species of Fusarium. But as I mentioned before, this Fusarium genetically is very different than the other Fusarium species that occur in the US. So the only close relatives of this Fusarium are endemic to China. And so it might have a very different mode and methods of functioning compared to the species that we're more, have more experience with. So ABG is using a paper that was written about fungicide applications to treat laurel wilt disease in Red Bay as a guide and to conduct a similar study with Torreya. And so they're just really beginning these trials. I haven't heard back about any progress on it yet, but hopefully soon we'll be getting some info from that. So as I mentioned, Tradekeepers is a collaborative project. We work closely and in conjunction with the Atlanta Botanical Garden to achieve our goals. And Ashlyn Smith, as I mentioned, is who I work with on the ground regularly. And this portion of conservation work would certainly not be, possible without our leaders on this effort, Emily Coffey and Juliette Renier, who has since left FMPS when she moved to Vermont. We're so sad, but they have done so much to spearhead this effort for conservation of this species. And I'm really grateful to them. Okay, so why survey private properties? Some people might ask, why bother surveying private properties for the species? Don't we have enough of them on public lands? And aren't they better protected on public lands? And isn't it a big hassle to, and a lot of resources used to contact private property owners and survey those areas, obtain permits, etc. And as many of you know, there are many reasons that we need to survey for all rare plants on private property, especially with a species that has become functionally extinct. Preserving the genetic diversity ex situ, having as many different genetic individuals in conservation outside of its native range is critical if we hope to reintroduce the species. And there is hope to reintroduce the species because there is a lot of hope in treating the Fusarium, because other Fusarium species do respond very well to fungicide applications, and can be kept in check or controlled with fungicide. So the more individuals that we have represented in conservation collections, the better chance the species has of recovering in the wild. And in 1986, the recovery plan for Torreya was issued, and it actually states that private property should be searched for Torreya trees and cooperation sought from owners to protect and allow access for conservation purposes. And also, these properties, we really want to get as many of these genetically separate individuals as we can, and they were hit just as hard by Hurricane Michael, and they're having all of the issues that are contributing to the decline of Torreya caused by Hurricane Michael. It's not just the initial tree loss of the canopy, but it's also erosion is really increasing because of the loss of so many mature trees. Erosion in the ravines has actually really increased a lot. In fact, just last week, I found one Torreya that was literally growing on a little cliff that was just probably will soon collapse underneath it. So because of erosion. So We collect cuttings from the trees that we find. And if they're healthy enough, we collect DNA from every single tree we find. And if they're healthy enough, we collect cuttings as well that will be grown out at Atlanta Botanical Garden. And the cuttings have to be taken in the fall and winter. There's a short, a few month long window of when you can take cuttings from this tree that will successfully root and grow. Otherwise, once it starts to getting warm, the cuttings will not, they will not take. So in the fall and winter of 2020 and 2021, we collected 73 cuttings on private property and 9 on public property. And these all represent newly found and numbered individuals. And the more genetic diversity represented in ex situ collections, the better chance the species has of survival. So, and this is a photo showing how difficult some of these surveys are when post Hurricane Michael because of this secondary growth. It's so dense and thick. Oh, a few more kind of illustrations of what this work is like. So on the left here, you'll see how steep the ravines are. Us kind of climbing up on our bellies. And then here's a sea of grapevines, which I did actually find a Torreya underneath that sea of grapevines. So it's not easy work to survey these ravines. It's definitely difficult. And then it gets hot really quickly. And I do survey into spring. I stopped surveying into summer because it becomes, there's so much leaves, so much foliage, the visibility is not good. And then it just becomes too hot and my volunteers are not happy about it. I'm gonna have to progress this way. I think, yeah, okay. Okay. So I've had a lot of the work is all volunteer based. I mean, I'm a paid employee, but I rely on volunteers for help. And I've had some really wonderful volunteers and I'm really grateful for them. So I just wanted to recognize some of them here. And so the 2020 last year's season was a really successful season. We surveyed over 600 acres, over about 16 different privately owned properties, and we found over 160 trees. Probably it was around 180 at the end of it. So that's a big. Oh. And so again, in 2019, 82% of the population of Torreya was known to be on publicly owned lands, and now it's only 66%. So as we continue, we're just really scratching the surface of surveying privately owned lands. So as we continue to survey, we might end up with more on privately owned lands, which is, it's really important to get all those genetic individuals. So again, here we are taking cuttings. There's Ashlyn with our little baby cutting wrapped up, and we got 73 cuttings from private property and 9 cuttings from public property. And so, sorry. We're all just having issues tonight. Okay. It keeps doing this. Sorry, I'm balancing 2 computers so I can look at my notes and and do the slideshow. Okay. So we collect cuttings from the trees we find if they're healthy enough for ex situ conservation at the Atlanta Botanical Garden Conservation Nursery, which is separate from their, you know, the big Atlanta Botanical Gardens in Atlanta. So these are some of the people from ABG who are working at the conservation nursery, and they're actually working right now to expand the nursery. And because we already have over 500, close to 700 Torreya trees represented in Atlanta Botanical Garden ex situ collections, that means that many different genetic individuals. So there are, Torreya taxifolia is represented in ex situ collections more than any other species in the Southeast. So it's not gonna go extinct because we have got our bases covered, luckily. And so these trees are not only backup for genetic material, but they're also seed for a project, for seed trial projects, which I'll discuss. And so again, as I mentioned, as the number of cuttings increases, because I keep collecting them and keep sending them up there, the need to store and care for the plants as they mature also increases. And so ABG has secured more funding to expand their nursery. And that's led by John Evans at the Atlanta Botanical Garden. Okay, so conservation genomics. This is another component of any rare plant conservation effort. It is not my area of expertise, but you all likely know that, again, as I mentioned many times, genetic diversity is strongly associated with the likelihood of species recovery. And so the ability of species to be able to resist disturbance and resiliency to recover from it is strongly tied to the diversity of the genetics represented in the species. So conservation genetics utilizes genetic theory and techniques to reduce the risk of extinction in threatened species. And for Torreya specifically, conservation genetics can be applied to allow us to know how genetically robust the populations in the wild are, as well as how successfully we have been at capturing genetic diversity in the ex situ collections. So we want to have as diverse genetics as possible in the collections. And again, to contribute to this work, we collect DNA samples from every single Torreya that we have found. And so this work is being carried out through a partnership between the Atlanta Botanical Garden and the University of Georgia. And below is a list of results thus far. But the last item on the list, developing a probe set to perform sequence capture on 325 Torreya individuals, is underway. So individuals found this year on private properties are going to be included in this analysis. So that's really exciting. So long-term monitoring is also part of the Tradekeepers project. We want to gather as much information as possible on how Torreya responds to disturbance like Hurricane Michael, and whether there will be more devastating long-term effects from the hurricane. So preliminary post-hurricane assessments, as I mentioned, revealed 20% mortality from Hurricane Michael, and 40 trees were affixed with humidity and temperature monitors and annual growth measurements, as well as ecological habitat assessments are conducted every year and will be for the next 10 years to provide information on how these trees are going to be responding and are responding to this disturbance. And Emily and Loy are heading those efforts at the Atlanta Botanical Garden.
Hey, Lily.
Yep.
Do you think we could wrap this up in a couple minutes so we could get to a couple questions, have time for some questions?
Yeah, I mean, do you want me to, well, I'm almost done, so
okay,
I can finish. Yeah. So we're also doing seed trials, which is basically planting out seeds in different areas and seeing how they do. And we're still collecting data on that. And then we also have habitat suitability models, which Nicole Zampieri, who she is an FSU student, and also she works for the Florida Natural Areas Inventory. She's she's a friend of mine, and she decided for one of her classes, she had to create a habitat suitability model. And she created one for Torreya, and that was very spot on. It was very useful. And now ABG is building upon that to figure out areas that would be best to survey, one, and then areas best for reintroduction. So I just wanted to recognize some volunteers. And of course, this work would not be possible without grants from the Florida Department of Agriculture and Consumer Services, as well as the U.S. Fish and Wildlife, and individual contributions from FNPS members and conservationists and supporters. So yes, that's it. Thank you. All right, you guys have questions? Should I stop sharing my screen?
The chat is now open.
Okay.
Yes, so does anyone have any questions for Lilly?
The nursery for ABG in that area, or is it in Atlanta?
The question was, is the nursery for was it APG in Atlanta, or is that up there in Florida?
No, the conservation nursery is outside of Atlanta in Georgia.
Got it.
As a mechanism to identify Chinese species that is resilient to the fungus, the sequencing of it, so it provides retrovirus to potentially attack into the genomics of ours.
Could you hear that?
I didn't hear that, no.
I have no idea how to repeat what you just asked. A very scientific question about, has anyone tapped into the genomics of?
If you have specific questions about the genes, genetic work, you can, I think I have some of the, I guess I don't. Oh yeah, here it says for Dr. Easterman, Lauren Easterman at the Atlanta Botanical Garden. That's who you would ask about the genetic work that they're doing. I do not, that is not my field.
Got it. A photo is being taken.
Okay, I will ask.
Any other questions from in here? We may have some in the chat. Let's check. Someone says, I have 4 trees planted in 2007 before the discovery of the Fusarium. Are Torreya keepers suggesting these trees be removed?
We're not suggesting that, no. I think this, it's so here is some info about the Fusarium. So planted trees are much less likely to show signs of infection. And also they're less likely to spread infection if they're not showing signs, because if fungus has to, create spores and grow. And so if you aren't seeing signs of the fungus on the tray, on your Torreya, then it is likely considered to be still infected, but is less likely to spread that infection if it's not showing outward signs. So, and also, it's already there. So we're not suggesting that anybody should remove them. We are just saying, we're just suggesting that planting them outside of its native range is putting other species at risk.
Got it. I'm sure that will make Fred happy.
Simple question.
All right.
If you want to volunteer, how will you volunteer?
If someone wanted to volunteer, how would they volunteer?
They can contact me, and yeah, I'm happy to have volunteers. You have to consider it's very physically taxing work, and you do have to be flexible even because we're crawling over and under a whole lot of felled trees. We're cutting our way through dense thickets of blackberries. We're climbing up these steep, flat, slippery ravines. It's not easy work, but if you're up for that kind of work, I enjoy work like that. So I like being outside, and it's kind of like being a kid in a jungle gym again. Although I do not like the blackberries at all. but yes, just contact me and I'd be happy to have I am happy to have volunteers. I even had a volunteer who she's a park ranger at the Everglades National Park, and she came all the way up here just to volunteer for a few days for Torreya surveys.
Nice. Yeah, and that's at TorreyaKeepers@fnps.org.
Yep, it's in the chat there.
Perfect. All right. Well, thank you so much.
Well, thanks for having me. I'm sorry we all had technology issues, but it's still fun to interact with some plant nerds. And I miss the interaction. I only get to see volunteers these days. So thanks for having me. And if there are any other questions that you guys have, you're welcome to email me. And I can put you in touch with, if I can't answer it, I can put you in touch with someone who can answer it. So. Yeah.
Perfect. Thank you so much.
Yeah, thank you guys. I'm stopping sharing, and are you guys closing down shop now? Should I just leave?
Yeah, I think they're closing. Good job, Lily. Thank you.
Thank you.
Right here.